Hi, Dr. Byrne. I have our first question for today. Put simply, what does the immune system have to do with gene therapy?
Well, Allison, good to see you today, and glad to have this conversation about the immunology behind gene therapy. So, we all know that in childhood, we try to prevent childhood illnesses with vaccination. Vaccines are intended to protect us from infections that can cause illness. And in the same way, we respond to other viruses that are acquired or that we experience naturally. So, in the context of an AAV treatment, the exposure to the virus, especially the protein portion of the virus, the outer capsid material, is what generates antibodies. And those antibodies are often very high and sustained over years and years, leading to the inability to retreat patients in the future. They also can be detected prior to exposure to a gene therapy treatment, and those preclude patients from being treated with that for that particular condition.
Can you define the most common virus used in gene therapy clinical studies?
Over the past 10 years, there's been a growing use of the form of parvovirus known as AAV, or adeno-associated virus. AAV is a small single-stranded virus that's very prevalent in nature. In fact, more than 50% of adults have been exposed to some form of AAV by environmental exposure. And these often travel in association with their helper virus infections like adenovirus, given the name, or herpes viruses are commonly associated with AAV infection. So it always is part of an infection with a more pathogenic virus like adenovirus or herpes virus, because AAV itself actually doesn't cause any primary disease, and that's why it was chosen as a vehicle for delivering therapeutic genes into cells.
What does a patient or caregiver need to understand about immune responses to an AAV gene therapy when considering potential treatment?
When patients are being evaluated for treatment with an AAV vector, one of the most important criteria to determine eligibility is the presence of pre-existing antibodies. Sometimes this antibody test will be by different names, depending on the sponsor and the product being tested. In all cases, actually, the AAV test for antibodies is specific to the product that's being used. The term total antibodies is most commonly used. That's a form of testing that looks at all types of antibodies against AAV. Sometimes they're specific to the one that is intended to be used, and sometimes they're non-specific or so-called cross-reactive antibodies. The other test is called a transduction inhibition assay, and this is how one determines what are called neutralizing antibodies. They're called neutralizing because they block the effect of the virus in infecting cells in a tissue culture dish. It often is thought that maybe the interactions in the body are a bit different than those interactions in the dish. That's what's led to this total antibody assay being more commonly used, at least for the current commercial AAV products. The total antibody assay is the preferred test.
Is there a specific threshold for antibody titers that would qualify somebody or exclude somebody from a clinical study? Or is it really dependent on the specific trial?
Yeah, that's a great question. So every product test, since they're different, requires the sponsor to determine what level of antibodies are permissible to allow the delivery of the treatment to be both safe and efficacious. So even though the antibodies might be slightly elevated in one scenario, that might eliminate the ability of the virus to get to the target if that is the musculature or the heart or the brain. The most commonly transduced, or where the genes are delivered after AAV transduction, is in the liver. So that might allow for slightly higher thresholds. In the two commercial products that are most commonly used for SMA and Duchenne, they have very different antibody titers. The product for SMA, Zolgensma, has an antibody titer of 1 to 50, which is almost at the lowest level that one can measure. So that is called the limit of detection. A higher threshold was established for the RH74 product that's known as Levitus. So there more patients would be eligible. We find for the Levitus product, for example, because we find that these track very closely together. If you do use a universal assay, which has been developed, it's just not used for commercial purposes, we see that all of them are related. This is an important thing to know because that means you can't just switch from one type to another to evade the antibodies that might have been developed after the first exposure.
How does the route of administration in different clinical studies really have an impact on the immune response and what level of pre-existing antibody may be tolerated?
So the very first studies were done with very minimal amounts of AAV in a space that's behind the retina called the subretinal space, where some of the important cells for vision are located. And that led to the development of the first approved product in 2017 that a very small exposure was anticipated because of the small amount of virus, and that unique space of the retina allowed Luxturna to be used consecutively from one eye and then subsequently the other one. We don't have the same situation in other compartments. They're like the systemic route of delivery has a much higher amount of the vector being given and therefore a greater immune response. So that's why in the systemic routes, like for Zolgensma, the second product approved in 2019, we know that patients develop high sustained antibodies that would prevent them from being retreated. Luckily, in SMA, the intended target are brain cells or neurons that don't divide over the lifespan. So when the vector enters into a neuron in the brain or spinal cord, it stays there for the life of that cell, and that generally is the life of the patient. So right now, we're passing 7 years of some of the first pre-symptomatic patients who were treated with the AAV9 product, Zolgensma, and it looks to have have a durable effect. So this is very encouraging, and of course, the clinicians who've treated patients over time, and there's almost 4,000 patients now, a degree of experience with Zolgensma around the world. We continue to monitor the effectiveness, but it's really been transformative in the care of patients with SMA. For muscle cells, it may be different, and that's because as we grow, the musculature grows, and as we muscle diseases that might have increased turnover or renewal of the muscle cells themselves don't receive the virus at the beginning. First exposure, so that may not last as long, but we suspect that even if there's some period of durability, this will improve function in patients during that time.
Now that an increasing number of diseases are being diagnosed by newborn screening, how does the maternal immune environment play a role in treating babies with AAV shortly after birth?
Important question, because sometimes there are misconceptions about this issue. As adults who are moms having children who might have a genetic disease, their exposures environmentally are the same to the rest of the adult population. And we know that up to half of them can have a very minimal titer to AAV, that during development, the antibodies of the mother are passed to the infant through the placenta. One of the common misconceptions is that babies acquired antibodies through breast milk, and there are certain types of antibodies that are transmitted and through breastfeeding, but they're not the ones that influence the ability to treat with AAV. And we have a recent publication about this where we examined a number of mother-baby pairs, looking at the breast milk antibody, the mother's antibody, and the baby's antibody, and determined that there's no detectable IgG, which is the type of antibody that's measured for eligibility of AAV, is present. And so we think that should not prohibit people from continuing to provide breastfeeding in those infants. That's not a risk factor for eligibility for treatment.
Great. Thank you for that new informative data. So for our next question, once given an AAV gene therapy, how can immune responses be managed?
Well, this has become exceedingly important in this era of higher dose therapies, trying to get either greater durability or a more widespread effect. And what's been observed in several studies is that we're really coming up on the peak amount that can be tolerated by the body. So these are known as innate immune responses. And in order to manage the innate immune responses, initially the thought was for hemophilia gene therapy that the only thing that was necessary to blunt those immune responses was to give steroids or glucocorticoids. And that was very effective for those lower dose therapies targeting the liver. Higher dose therapies targeting the brain, or used now in bigger populations where we have more examples of adverse events, have led us to believe that the antibodies that form within days after exposure, so often exceeding within even 48 hours the ones that are present prior to the treatment, can cause a safety issue, particularly as it relates to one part of the protection that we have against pathogens called complement. So the complement proteins protect against other kinds of invasions and help damage the cells that might be in an infection, and they can damage our own cells. Is a bystander effect. So we've learned that in some cases where a certain type of vector is used at a total amount, for example, maybe in adult patients where they're larger and the total exposure is high, it's become necessary to block the antibody responses. So we can do that with some existing drugs that prevent B cells, which are the ones that make antibodies, from being present when the vector is given. This helps the vector do its part in the first 2 weeks after exposure because there's a delay in the onset of the antibody response, sometimes up to months of delay, and that actually helps the given dose to be more effective because it stays around longer. And we're additionally trying to develop more specific ways to inhibit only the AAV antibodies and not all antibodies. This would help keep patients going back to school and not worrying about other infections or having any form of immune suppression, which is really only transient in this case. It's not a permanent effect, really during the first month. But that's the strategy that's being used. And then we continue to monitor for later effects of immune activation against the protein that's being made, the transgene, or the late effects of the capsid, particularly in liver toxicity. So that's why later follow-up studies are done, and other drugs can be used to reverse those effects even after the treatment's been given.
Is there one specific immune modulation protocol that's used in all trials, or does it vary depending on the product?
It really varies depending on the product. Some sponsors have felt that it's sufficient to use the glucocorticoid treatments around the time of dosing and then slowly taper those off. I think as we learn of the susceptibility of some individuals over others, we'll be able to use some pre-screening techniques, particularly that look at specific genes that influence immune response, and use more aggressive immune management strategies to those who have higher susceptibility. Of course, there's a strong interest in being able to treat patients a subsequent time or add to the effect that they might have had in initial exposure. Those will require much more comprehensive, ideally preventative strategies towards antibodies, but that group will also require kind of special attention.
How does the immune response to AAV impact treatment outcomes or the potential to get a subsequent dose?
Yeah, as I mentioned, we have a wide range of responses. If you were to look at the immune response in the population of everyone who got their tetanus shot when they were little, it is all over the place. And we know that at least in those cases, that doesn't really last forever. That's why we get booster shots. What has been learned about AAV immune response is there's a certain threshold, which is a very low amount of the vector exposure when given systemically, and those antibody responses seem to last a very long time. In one individual who got a local administration of AAV now 25 years ago, the very first muscular dystrophy patient with limb-girdle muscular dystrophy 2D, he's still antibody positive 25 years later. So we don't expect these to just go away on their own. We're actively looking at ways to use the immune system itself to eliminate those immune cells, and along with preventative strategies to lessen or completely eliminate the immune response. So I do think this will become possible. It's just going to be important to have a rationale for in what circumstances is it needed, and what risk tolerance do we have for that notion of treatment that is, I think, almost certainly to come in the future.
Sounds like there's a lot of ongoing research to both modify the actual AAV virus to lower the immunogenicity of the treatment, as well as improved immune modulation to attenuate the body's response to the virus. Is there something particular that you're most excited about in this field?
Well, I'm very excited about that notion that we can lessen the overall exposure, particularly related to these innate immune responses. It was the lifelong goal of Mavis McKenna, who's one of our faculty here at University of Florida, solve the structure of AAV that enabled researchers in the current day to use that map of the surface to add specific ligands or higher affinity receptors for different cell types in the body. This has been done for the brain, for the musculature. There are now tropic vectors for different types of brain cells, the kidney, the liver. So this is, I think, the next wave of excitement for the AAV field. These products are probably going to be a little harder to manufacture, but I think it's going to be worth it to solve those issues and improve the efficacy and lower the overall exposure, and hopefully increase the safety of AAV gene therapy going forward.
How does the quality of the AAV product play into the immune response?
That is a really important point. I'm glad you brought that up, because there is a group that's developing a reference standard for AAV materials that'll help us compare one product to another. At our group, we did this many years ago with AAV2 when it was the only one in use for local regional administration in the eye and the brain. Now we need a new one, utilizing the new methods and analytical techniques that are available to better characterize AAV. When those suite of tests are available, it'll help sponsors and investigators, we hope worldwide, characterize their products to the highest level of quality, because other materials, other biological materials, DNA and proteins that are co-purified with the virus can influence the immune response. And of course, we can't eliminate the most active part, the capsid and its genome, but we know that we can eliminate parts of the vector material that have only part of the genome, they're not helpful, or that have no genome at all, so-called empty particles. These only increase immunogenicity and lower the efficacy. So new analytical techniques and better scale of production will enable us to get more purified products that will be safer and hopefully more effective.
Great, thank you. Okay, last question for you. How are animal studies used in preclinical research to better understand immune responses to AAV gene therapy?
Well, characteristically, mice are used as models of given diseases because for many, many years, there's been an ability to modulate the immune responses in mice by understanding all the genes involved in immunity and in the given disease they represent. So combining those things is a very powerful tool. Of course, they don't exactly replicate the human immune system, but I think there's an opportunity as we develop more specific capsids for certain human receptors. Those can be used in mice to improve targeting so that we can study, just like in the human context, these lower doses of vectors that match the human receptor that they're targeted to in a mouse. And that mouse could even be designed to have the underlying disease that's being studied. So this would potentially lessen or eliminate the need for larger species studies, which add cost and time and are not so far been that representative of the human immune responses. So as we learn more from existing clinical products and products in development, use more sophisticated assays for the immune response and done early after delivery. This is one of the things that we missed some of the early time points in studies that were before we understood these events. Then I think we'll come up with even better ways to mitigate the immune response and improve the outcomes.
In addition to murine models, what is the utility of using larger animal species? But then also, where do those fall short when trying to study the immune response to gene therapy?
Yeah, that's an important point. I think in some animals and in some cases, there was a need from regulators to mimic as closely the human immune responses in primates. Primates have evolved some protections against the common viruses that impact them in the way they live in an unsanitary setting. So they have attenuated their response to viruses in order to tolerate them more. And that, I think, was one of the reasons why we didn't see the magnitude of immune responses that we do in humans. Other species that are domesticated, like in cats or canines that have a more hygienic environment, they do respond more like humans to infections. And there are many ways in which we could study those as well, but they're harder to manipulate in terms of making disease models. So we kind of wind up back at rodents again, and that's where I think we have the opportunity to mix specific receptors from human cells where vectors are being targeted into the mouse and then studied in the context of the disease that can be created, whether it's a dominant or a recessive disorder. The mouse model generally exists.
Thank you very much, Dr. Byrne. I've really enjoyed our conversation today. Thank you for lending your expertise on AAV immunogenicity for our community quick takes.
Thanks so much, Allison. And of course, I'm sure you and I are both looking forward to attending the ASGCT meeting in New Orleans. So we'll see you there.
Absolutely. See you in May.